First of all, thank you to Danielle and Julian for inviting me. It's always been very inspiring. I'm quite a person that likes to come to Café Scientifique, and I always like the questions. They're always very inspiring, and I'm sure that I will have plenty of questions afterwards which actually might inspire new creative ways to deal with my research. Now, I study and I research actively the immune system, which is essentially a system that defends ourselves against invading pathogenic organisms, but it also protects us on a daily basis against cancer growth and cancer development. So why is it really important to study the immune system? Well, from a historical perspective, can you hear me at the back?
Yes.
From a historical perspective, protective therapies, for example, vaccines, have proved extremely important in the eradication and the management of pathogenic organisms such as mumps, measles, rubella, polio, and so on and so forth. And that was really the first way scientists started looking at modifying or modulating the immune system so that we can really counteract these pathogenic infections. More recently, we are now looking at cancer immunotherapies as a new weapon in our fight against cancer. And don't take my word for it. This is a movie here, and what we are seeing here are white blood cells shown in these yellow dots. We label them with a certain color so that we can distinguish them from other cells. These grey cells are healthy cells. These blue cells, however, are the cancer cells. And now what you will see is that these serial killers, so these serial killer immune white blood cells, will swarm around. They will attach to the cancer cells. They will also attach to the healthy cells. They will scan them. They will scan both. And they will kill targetedly and selectively only the cancer cells. That is why we need to start looking at how to use the immune system and bolster the immune system to keep on attacking the cancer cells. So you're seeing a swarm of these yellow dots. And each time you see a green signal, it means that the cancer cell was killed. So a cancer cell was killed there. Now look here, there's a cancer cell there. You will see a yellow killer cell coming on, and there, it kills it. It's not killing any of the healthy cell, and that is very important. Why? Because the most important thing in cancer therapies is that we leave the healthy cells untouched, but selectively target the cancer cells. So, I am going to discuss briefly about what the immune system is. We can divide the immune system into two tiers. The innate immune system, which essentially offers a barrier to invasion and attachment of pathogenic organisms, and then we have an adaptive immune system, which is a system which modifies through cellular mechanisms and develops a way of attacking the pathogenic infection. So, there needs to be communication between the innate and the adaptive. Why? Because the innate immune system is capable of recognizing things that belong to the self, so things that belong to us, things that are healthy, but also discriminates self between non-self. Non-self being pathogenic organisms or cancer cells. The adaptive immune system cannot do that. The adaptive immune system is essentially there as a system to attack pathogens that evade the innate immune system. And the process that links those two tiers, so the innate and the adaptive, that process is called inflammation. Inflammation is actually a process that was discovered by the ancient Greeks, which essentially means put on fire, which is what you get when you have a wound. or you have a glow, you have a bit of heat, so it's on fire. You have pain, you have swelling. And the thing with swelling is it's critical. Why? Because there's white blood cells within it of me cutting my skin, for example. There will be pathogenic bacteria, pathogenic viruses, or pathogenic fungi that will be invading my body. There will be a swarm of white blood cells, as I will show you in this movie here. So that here is a blood vessel. Those green dots there, those green blurbs, are cells, white blood cells, which we labeled green, but they're white blood cells of the innate immune system, and they are coating the lines of the vessel. And they will swarm into where the infection is. So the infection is roughly around here, where you see this grey here. And they will spread through. And that is pretty much accounts for the hallmarks that I mentioned of inflammation. Swelling, heat. And you see this incredible flow of these white blood cells into the infected site. Now, let's go back to my paradigm of slitting, having an injury. Danielle is going to help me a bit here. So a critical cell that acts as a communicator, and Danielle is a very good communicator as well, so that's great, between the innate and the adaptive immune response is called an antigen-presenting cell. I'll explain what that means. So, Danielle is a professional antigen-presenting cell. It's a dendritic cell as well, called a dendritic cell. So, if we have, for example, representing a virus with a flower, so it evaded the innate immune response, right? So it is multiplying. It's growing. It's can cause major infection. Danielle is the dendritic cell. I am a part of the adaptive immune response. So I'm one of those adaptive immune cells that cannot recognize the virus. I am, by the way, called a T cell. Actually, I'm wearing white, which is great. And you're wearing some of that, which is great. So, this is the virus. You'll realize that that's a virus. Okay. What Danielle will do, but she won't do it, is that she will eat it. And that means that she is phagocyting the flower. And then she will present, if you cut a petal off.
I'm sorry, I have to.
So she'll present it. You remember I mentioned she's an antigen-presenting cell. This is the antigen. She won't present the whole virus. She will present part of the virus. She will then come to certain vessels called the lymphatic system, into a lymph node. And there she will meet cells of the adaptive immune system, such as myself, I'm a T cell. Now, there are various repertoires of T cells that are responsible for recognizing viruses, T cells that are responsible for recognizing bacteria or fungi. But I am specific, I'm a naive, but I'm specific for that virus, for that antigen. So I have certain receptors in my hands. I will go, oh, hello, there's that antigen, and I will form a hug. And this is the immunological synapse. This is the interaction between an antigen-presenting cell and, until now, a naive T cell. There are around 1 in 100,000 T cells like me right now. But over the course of infection, after this interaction, I will have signaling events going on through my cell, and I will start proliferating, start growing. From 1 in 100,000, we will go to 10% of the population. So we'll go to 10,000 per 100,000 T cells in the course of about 5 days of infection.
So do you keep this?
No, you actually keep it.
Ah.
I don't. You always. You always, because you will be presenting it to many other T cells. If I didn't have any interest, I wouldn't interact with you. I only have an interest in you, and actually, the higher the interest, the better, because that means I would proliferate faster. So that is the course, that is called an immunological synapse. And just to show what a part of an innate cell, they actually chase actively the bacteria. In this case, it's a bacteria. And this is again a cell of the immune system. It's really quite interesting. So you see that that's the bacterium. Part of the cell, innate immune cell, is actually chasing that bacterium until it phagocytes it and kills it. That's the innate. But then the whole process that I referred to just now is called the communication between the innate and the adaptive immune system. Now, that's in the context of microorganisms. What happens in the context of cancer? So we have other cell types in the immune system, which are also T cells, but we call them killer T cells. And these are serial killers. They specifically swarm around the body. There are around 10,000 of, more than 10,000 actually. There are about 15 million, actually, sorry, that's the wrong number. 15 million per teaspoon of blood. And these move around quite actively in the tissues, seeking the telltale signs of cancer. So you see they have these amorphous blobs moving around, and they move along by performing this leading edge. And they scan and scan and scan. And that's important. These are the only cells in the body that are motile, by the way. The only cells in the body that are motile are these immune cells. They will seek a cancer cell, shown in blue, and through membrane protrusions, similar to my hands earlier, they will interact with the cancer cell. This is engaging a cancer cell, and what you're seeing is these red blobs are actually the killing molecules. They will migrate along specialized structures called microtubules, go to the interface, which is called the immunological synapse. They will puncture the surface of the cell, injecting the lethal poison. This is again another image of a cancer cell. This is a cancer cell. This is a T cell that is injecting specifically and killing it. What's important from this image is see that's killing the cancer cell again, but it does not modify it. It just move on. It killed it and then move on to find another target. And really, the immunological synapse can, if you wish, parallel, if it works, the neuronal synapse. Hold on a second. Yeah, okay. Great. So it parallels the neuronal synapse. Most people might have heard of the neuronal synapse. So a synapse that is a neuronal is essentially a way in which electrical signals are transmitted from one nerve cell to another, or from the nervous system to the muscular junction. This is how I manage to flex my hand, and that is how the messages, the electrical signals, are moved from one nerve cell to another. This is essentially the same thing. You have a bullseye structure in the immunological synapse, which is essentially where the antigen is on the antigen-presenting cell. It is formed inside the center. But let's look at it, how it looks at it in real life. This is actually an experiment I have done, provided it works. Yes. Back in the UK, I actually did it. It was probably about 3 o'clock in the morning, and I was quite surprised. And everybody was probably having some whiskies. This was a Friday night when I spent the time in the dark room, just looking at cells. It can be very exciting. So these are immune cells, right? These are my cells of interest. What I did is I take a glass slide, and I coat that glass slide with the membrane, so the outer surface of the cell. In the case, it would be similar to what Danielle's skin is, but it is essentially the surface of the cell. So these cells are actually interacting with that membrane. And let's play a movie. So you see how they're forming those membrane protrusions. You can see the membrane protrusions. That's because it's interacting, because there's the antigen coated on that slide. That one is not interacting at all. It's just still searching around, still looking around, still looking, still looking. But what is actually happening under the surface? So, and that slide was actually coated with certain proteins. So the proteins that are involved in the signaling events, right? So what you will see are the proteins that will be recruited by that cell, and that are forming adhesion, and they will form that bullseye structure that I referred to. And in cyan, you'll see those molecules moving centripetally. And that's really important to study the dynamics of this immunological synapse. It's really important because that's how we can develop new therapeutics that enhance that formation of that structure, therefore enhancing the delivery of the poison in these cytotoxic or in these killer cells. And it's 3 o'clock in the morning, and when I was doing this, but I was still quite jumping about all over the place. And it is a beautiful image when you see something like that happening. So this cell is just doing it out of its own accord. There's nothing, it's just moving these signaling molecules centripetally. And what we found also, this was done with a friend of mine, is, remember I mentioned there's this bullseye structure. So it's really important that these molecules, these granules that kill cancer cells are really focused, right? Really focused towards the center of the synapse. So in this case here, I didn't add a certain molecule on the surface of the glass. And you see that they are somewhat distributed evenly across the surface. And I'll play that. You see how these red dots are kind of uniformly distributed. They're not polarized, they're not targeted. That's not really good for the killing activity of the cell. But by adding just one receptor that specifically causes the recruitment, we see that it is really polarized. If that's the surface of a cancer cell, and that's our killer cell, that's exactly what we want. That's the therapy that we want. We want targeted delivery of the poison to the cancer cell. And from then, I decided to, okay, how am I going to shift from a glass surface to look at how to identify what other molecules are involved in this structure. And I remember my mentor was saying, oh, why don't you just take the glass slide, remove all the cells, and then take the glass slide and just look at that. And I was just like, that's insane, that's going to take me ages. So I said, no, I'm going to try something else. Well, behind his back, really, because after work I kept doing this. So I said, okay, I'm going to develop a different system. So I took glass beads, roughly the size of the cells, and I coated them with the same membrane of an antigen-presenting cell. And I was able to reconstruct the immunological synapse with the killer T cells. And what you see there is essentially a reconstructed. So this is the killer cell. It's not playing. Oh, it's not playing. Great. Anyway, so you would have seen funky of 3 dimension, but ultimately what's great about it is then I can separate these beads of interest and look at new targets that we can target therapeutically. And this is where my research is going to at the moment. So, I will be happy to take your questions. If you are interested, where is this leading to? So if we know about the surface of the cell and how they interact in the molecular determinants for the formation of this immunological synapse, we can actually start developing new technologies. This is CAR T cells, which is essentially a modification of our cancer killer cells. Here you have a normal cell, and this is a CAR T cell. So what one would do, and this has just been, I think, in the past few weeks, has just been approved. You take the cells from a patient, you actually put a receptor that is called a chimeric antigen receptor. It's coming up. Yeah, and that's the receptor there. That would be able to bind so we multiply them, and that would be able to bind. These are actually leukemias, so that's why you're seeing those. So it can bind with the cancer cell of interest and inject this poison. My research is essentially looking at new antigens and new receptors, and that's how we can develop new therapies for new other tumours, not just leukemias, which are blood cancers, but maybe even solid tumours. So I'm happy to take your questions. Thank you.